First, are there any questions or concerns? We will definitely finish chapter 3 today. We will start chapter 7, and your first exam is open. It's under this exams module right here. Also, your second lab practical is open. It will close in about 2 days. I'll extend that one out.
What chapters is that actually on? The lab quest 2.
Which one? The lab quest 2. It's over the lab exercises that we've done since the last lab quest. If you give me a minute, I'll look and I'll tell you specifically which ones. Give me 1 second. I'll tell you which ones it's over. Okay, so the second lab quest is over lab exercises 5 and 6, all the parts of 6. So 5 was on the chimney transport across the plasma membrane. And lab exercises 6 were parts A and B and C and D. Yeah, I'm going to extend it out. I'm not going to extend it out till next Wednesday, but I will extend it out until next Monday. The reason is I want to include it in the midterm grades that I have to submit in the next week. Your first exam, like I said, it's open. It's open until, I think I said the 20th. Come on, look. Yeah, October 20th. So a week from Wednesday, tomorrow. Other questions or concerns?
Can you repeat the lab quest 2 chapters again?
Over lab exercise 5 and lab exercises 6, all the parts of 6. Remember, we split it into 2 weeks, so it's over both weeks. Other questions? So I want to finish chapter 3 and then start finish chapter 3, start. Oh, I'm tired. I apologize. Give me a second here. Finish chapter 3, start chapter 7. At the end of chapter 7, or towards the end of chapter 7, which we will finish today. We'll finish today and next, we'll start it today and we should finish it next Tuesday. And then we will go from there. Your first case study, if you look at the syllabus, you have 3 case studies. I planned on opening the last case study, the first case study, this last week, but I wanted to cover the bone. And so I'm not going to open it yet. And I'll put out an announcement when I open it, but it'll probably be sometime this weekend. Watch your grades. I'm gonna stop sharing just for a moment, because if you go into grades on D2L, your calculated grade that it gives you is just for the assignments that you've turned in. If the deadline has not passed yet, as soon as the deadline passes, or within hopefully the next week or so, I go in and I put zeros in for everyone who hasn't done that. Because that's the way to calculate your grade. Is if you don't have zeros in there, it just gives you your grade for whatever you've submitted, whether you've submitted everything or not. So for example, your grade may be a 90%, but if you are missing things, it may calculate your grade to be a 90%, but if you are missing things, that might not be your grade. That's just what D2L is submitting for what your grade, for what you've submitted. That's what D2L is calculated based on what you've submitted. So for right now, you should have all of chapter 1, the lab practice, the smart book assignment, all that, and the quiz, all 3 of those done for chapter 1. You should have all 3 done for chapter 2. And you should have at least a smart book assignment for chapter 3 done. And by next week, you should have the practice, the smart book assignment, and the chapter 1 quiz, chapter 3 quiz, all done for chapter 3. You should also look at the syllabus and make sure that you've done all of the virtual labs that are listed for up through now, because we're definitely on schedule for the lab portion of this class. Should also make sure that you've done all the parts of the homework for the labs. And I know that there's a lot of people who are falling behind. That's why I'm bringing this up. Because your calculated grade for your midterms is going to include chapter 1 through chapter 3. And it will include all of the lab exercises up until now. All through the lab exercise 7 that we did last week. With this in mind, don't start the lab exercises for 7 yet. Not 7, 8. Lab exercises for 8, 9, and 10. I think they're a little long, and I'm going to take some out. I am going to make your lab exercises long in general, because a lot of what we're going to learn in a lab is anatomy, and anatomy has a lot of detail to it. And the best way that I know to help you learn the detail is to give you lots of lab exercises. And anatomy takes a lot of looking at it and a lot of repetition to learn it. If you already know it, it'll be really fast. But if you're still learning it, like many of you are, it's going to take time. So you'll see that the lab exercises, especially for this week, are really quite long. I am going to take some of them out, but there's 3 parts to it, and each part is fairly long. But it's all anatomy. And therefore there's a lot to anatomy, and there's a lot that you need to know for anatomy. So that's why the lab exercises are longer. Any questions or concerns? I know we have at least 2 people who have not taken the first lab quest. You need to get a hold of me and let me know when you're going to take it. And I'll extend it out until when you say you're going to have it taken by. Any questions, issues? So I just, so that you know, one more piece of information. Yes, I do submit midterm grades. Are they counted on your permanent record with AC? No, they are not. They are just a a way for you to measure accurately what your grade is at mid-semester.
Professor, I do have a question.
Go ahead.
Which order, like what should we do first between the lab quest, the quiz, and the like whatever the test? Because I feel like I'm doing things backwards. I feel like because there's one of them that's like, do you want to learn more about this subject before you go on to finish it? And
the smart book assignment, let me explain. The smart book assignments are there for you to read. Most of you, the majority of you, if we never had those smart book assignments, would not read the book.
Okay.
And what that is to essentially an incentive to get you to read the book. And you can see we cover a lot of information in this class, but it's only a drop in the bucket for how much is in anatomy and physiology.
So is that McGraw-Hill or is that?
It is.
McGraw-Hill, okay.
And so if I were you, right now, depending on where you're at, and let me think for a second. If I were you, and if you're worried about your midterm grade, I would get the first 2 lab quests done. If you haven't done the first one, get it done. If you haven't done the second one, get it done.
So lab quest.
Bear with me just for a second. Well, oh yeah, get the lab quest done. Then I would worry about the chapter homework, maybe the exam first. But if you have been doing the chapter homework alone, you're getting ready for that exam. That's the reason you're given that material in between the exams.
Okay, thank you.
You're welcome.
Is
But
lab
but the things that are going to carry the most weight, even though if you look on the syllabus, and let me go back to sharing my screen really quick. I'm going to spend a lot of time, hopefully not as little time as I have to, but I tend to have to spend a lot more time in online classes, reminding you to get the things that you've been assigned done, because people tend to get behind. And because it's an online class, people are less likely to attend, less likely to get the work done. And I hate failing students. But if you don't get the work done, I will fail you. But that's my job as a professor. I don't like holding students back. I don't like failing because I know it's holding you back. However, if you're not getting the work done, then I don't have a choice but to fail you. That's why I'm asking you to get it done. If you go to the syllabus will tell you how much each thing is worth. So if I come in here, the smart book assignments altogether are worth about 15% of your grade. The quizzes are worth about 10% of your grade altogether. The exams are worth about a 20th altogether in total of your grade, so about 20%. Your lab quests are worth about 15% of your grade in total. The virtual labs altogether are worth about, or what is that? About 1/15 of your grade. All the little tiny things, if you think that you do not do well on, if you don't think that you test very well, or you struggle with test grades, all the smart book reading assignments, all the chapter quizzes, all of the chapter practices, all of the lab work, the lab material, all of the virtual labs are all buffering your grade. If someone theoretically only took the exams and the lab quests and the quizzes, they would still fail a class. Because if you look here, they're only worth, let's see here, altogether. So lab exams are worth 300 points altogether. The chapter quizzes are worth 150, so that's 450. The final exam is worth 150, so that's 600. And the lab quests are worth 200 together, so that's 700, 800. So altogether, that would put them at about a D if they got 100% on all that. So I need you to make, you have to make a choice. You're in school. You take responsibility for your education. If you want to do well, get the work done. I understand that you have other life responsibilities. I do too. But I do understand what it's like to be a student. I was a student for longer than I ever want to admit to. But you do need to take responsibility for your education. And that's all I'm going to say on it. But if you stay up in the class, you're going to learn a lot. But your class is really, at the same time, the class is what you put into it. If you want to learn a lot, you put a lot into it. If you don't care one way or another, you don't put anything into it. But you get what you put into it back. And I know a lot of you, I'm preaching to the choir here. But I need you to make the choice. Many of you are doing really well. I'm talking with those students who are missing several things or have not done a lab quest. I need you to get it done. If you're in school, you do have deadlines that you have to meet. And in an online class, I do extend things out. However, you still need to get it done. Attendance, if you look here, is only 60 points altogether. So please get the other things done.
What did you say about the chapter 7 quiz? Don't do it yet.
Don't do the chapter 7. Do the chapters. Chapter 7. You can do it now, but I wouldn't do it because we haven't even started it.
Okay.
It's open to you, but I wouldn't do it. Right now, make sure you have everything done for chapters 1 through 3. Make sure you take the exam. Make sure you have the lab quest done. Make sure you have all of the lab exercises completed up through, as of right now, up through last week. And then on Thursday, we'll look at chapter exercises 8, 9, and 10. Okay, I'm gonna jump into class because I don't want to put more time into this. So centrioles, they are vital for cell division. They are composed of a set of pinwheels, look like a pinwheel, with 9 triplets of microtubules. They're vital for cell division because they're involved in the formation of the mitotic spindle. If you remember from lab, this is where the spindle has to attach to the centromeres of the individual chromosome homologs. That's why the centrioles are so important. There's a lot of anti-cancer drugs that are specifically target the mitotic spindles. Because if you can stop the cell from dividing, you can stop the cancer from growing. They tend to make people, most cancer drugs make people sick because they're not specific to just the cancer cells. They go after any cell that's quickly dividing, which your body is constantly recovering from injuries. Therefore, your cells are constantly dividing for that reason. So many cancer drugs make people sick because for that reason. Your hair, your fingernails, your skin cells that tend to be actively dividing all the time are affected by cancer drugs also. In addition to forming mitotic spindles, the centrioles also form the basal bodies of cilia and flagella. We have cells with some cilia. We talked about the ciliated columnar simple epithelium. And then we talked about, we talked about the epithelium in the intestinal tract and how it's mainly to increase the surface area. on a cell. The centrioles are part of the formation of the cilia. They're part of the formation of the flagella. We only have one animal cell that has a flagellum. Do you know what it is?
Sperm.
Yep. So cilia are whip-like multicellular extensions occurring in large numbers on cellular surfaces. They help with the flow of mucus and helping things to flow up sometimes and flow down in other ways. That you find them in the in the fallopian tubes, in the trachea. When the central projections are significantly longer, they're called flagella. Cilia propel substances across the cell surface. Flagellum propel the cell itself. There's patterns of microtubules in the cilium and flagellum. So in the case of the cilium and flagellum, you still have sets of microtubules. Remember in the centrioles, there's 3 triplets. 9 triplets in the cilia and the flagellum towards the base, especially towards the base of the flagellum, right here. Rather than having 9 triplets, there's now 9 pairs of doublets with a central pair in the middle. Extending from the microtubule doublets are arms composed of dynein. These are motor proteins that move along the microtubules, and as they travel along the microtubule, they pull on the adjacent microtubules, causing the whole tubule to bend. Just for a second. I'll put up this link for you in a moment. I only found this link the other day because I was teaching about the cilia in another class. So to cilia. Sure, I'm spelling it right. Let's do this.
Motocilia and flagella are hair-like
Sorry.
appendages of cells that generate fluid flow along tissues or allow cells to move through their environment. To better understand how these appendages generate motility, we flash froze sea urchin sperm cells while their flagella were actively beating, and then directly visualized them.
So here's your 9 doublets. 8 on the outside, 2 on the inside. And if you look in here, there's little motor proteins that are connecting one set doublet with the next doublet.
Molecular machines inside the flagella through cryo-electron tomography. Thousands of motor proteins called dynein.
So here's all the little dynein that are connecting one with the next.
Are arranged regularly along the microtubule scaffold. Through classification, we identified several different functional states of the dyneins as they step along the microtubules. We demonstrate a detailed stepping mechanism. From the relaxed post-power stroke state, the dyneins release from the microtubule, swing forward, and rebind to the microtubule. During the power stroke, the cart.
So it stays attached to one of them, one of the doublets, and reconnects, and it reattaches, connects, detaches, reattaches, detaches, reattaches.
Cargo is pulled forward by the action of dynein. As long as ATP is present, dyneins continue to walk along the microtubule. However,
and we've talked about ATP, that's the cell's currency for energy.
If dyneins receive inhibitory signals, they can be kept in intermediate states that resemble the post-power stroke state, leaving them unable to generate motion. Our direct visualization of specific dynein conformations on either side of the flagella suggests a switch inhibition mechanism generates the flagellar beat.
And that's all we need to see in this. Actually, let me see.
Molecular linkers resist this sliding, which causes the flagellum to bend in one direction. By periodically switching the side of dynein inhibition, the flagellum alternates the direction of bending, resulting in the typical whip-like motion. The propagation of dynein inhibition in different regions of the flagellar wave along the length of the flagellum leads to the characteristic.
So there's a pattern to it. So the crawling of the dynein along the doublets results in the cilium bending. There's a bending pattern that results in a propulsive power stroke followed by a recovery stroke. The cilium create a pushing motion in a single direction that's thus repeated 10 to 20 times per second. The bending in one cilium is followed by the bending of the other, resulting in this traveling wave. Why do we care about cilia? Well, you probably have heard about it if you haven't ever really thought about it, because we've all heard the smoker's cough, right? We know people who smoke and have a smoker's cough. Smoking, the reason for the cough is that smoking reduces the number of cilia. So there's fewer cilia to clean your lungs. For that reason, the lungs and the airways develop more mucus in order to try to keep themselves clean. Because the mucus is not cleared out well because there's reduced cilia, it often will stay in the airways, clog them, and make individuals cough. Because it's not actively flowing and being cleared all the time, smokers tend to cough. Because cilia also line the fallopian tubes and helps to move the egg cell towards the uterus, women who smoke have an increased risk of ectopic pregnancies, which is where the fertilized zygote embryo implants somewhere besides the uterus. The primary function for microtubules inside a cell is what?
B?
Oh, you're right, it's B, Miranda. The nucleus is the gene-containing control center for the cell. It contains all of the instructions needed to build up almost all of the body's proteins. It dictates the kinds and amounts of proteins that are needed to be synthesized in response to any given signal or any combination of signals. So it determines what proteins will be expressed by the genes at any given time. Cells can be uninucleate, meaning that they have one nuclei per cell. They can be multinucleate, meaning that they have multiple nuclei per cell. And when we talk about muscle towards the end of the semester, you'll learn exactly why that's the case. And that's because the muscle cells, several muscle cells will fuse together to form one fiber. And so all of the nuclei for that cell are now contained within one cell, which is what we term the muscle fiber. Anucleate, meaning that some cells don't contain a nucleus. Your mature red blood cells don't contain a nucleus. These cells, red blood cells, because they cannot create mRNA, they can't replace proteins that are deteriorating. And they also can't be reproduced or be replenished. They can't reproduce and replenish themselves. Some recognizable structures within the nucleus are the nuclear envelope, the nucleoli, and the chromatin. There's several other distinct compartments that are rich in specific sets of proteins that are in constant dynamic flux. But the three main ones are the nuclear envelope, the nucleoli, and the chromatin. The nuclear envelope, which, if you remember, it's continuous with the membrane of the endoplasmic reticulum. Has an outer nuclear membrane and an inner nuclear membrane. The inner nuclear membrane is like the terminal web that lines the plasma membrane, and that it helps maintain the shape of the nucleus. It also helps organize the DNA in the nucleus. You see there's chromatin-associated proteins. The nucleus, like the plasma membrane, is selectively permeable. So it allows some things to pass through it more easily than other things. However, in contrast, the nuclear envelope is more permeable than the plasma membrane. There's two layers of the envelope that interconnect to form the nuclear pores. Protein complexes line each nuclear pore and create a channel regulating large particles that enter in and out of the nucleus. So just like channels within the plasma membrane, we have nuclear pores that are channels. Via energy-dependent mechanisms, so this isn't passive diffusion, molecules are imported from the cytoplasm and RNA molecules and are exported from the nucleus via the nuclear pores. So RNA molecules, ribosomal subunits, all have to pass through the nuclear pores. The nucleoli typically contain 1 or 2 nuclei per nucleus. They're associated with nuclear organizer regions, which are the regions where the ribosomal RNA is synthesized and the ribosomal proteins for the ribosomal subunits are created. Most of these ribosomal subunits, like I mentioned in the last slide, are going to exit through the nuclear pores. I can't think of any that are actually don't exit the nucleus because protein synthesis occurs out in the cytoplasm. I'd have to read into it a little bit more to determine just why they would stay in the nucleus, unless there's some protein expression that happens in here, and that's not the case in eukaryotes. The nucleus contains chromatin and nucleosomes. When a transmission scanning electron microscope is used to look at the DNA and the chromatin, what the result looks like a string of beads that contains 30% DNA, 60% globular histones, and 10% RNA chains. Histones are these little proteins that, as an octamer, meaning 8 histones, wrap around the DNA double helix twice, or the DNA double helix wraps around the octamer twice. This octamer histone core with the DNA wrapped around it twice is called a nucleosome. Each nucleosome is connected to the next nucleosome via linking DNA. So these histones are proteins that they're a little underrated. We're still learning about their significance. However, we know that they're really important because they play a major role in regulating gene expression. When the histones are methylated, that are wrapped, in which the DNA are wrapped around, that DNA is silent for gene expression. It's not expressed. When rather than adding a methyl functional group to the histone tails, but an acetyl group is added instead, the DNA is now open for expression and is easily frequently expressed. Now, my question for you is, thinking back to chapter 2 and the functional groups, what do you know about the methyl functional group? And what do you know about the phosphate backbone that makes up the DNA? Someone go back and look. Tell me what you know about those two functional groups, the methyl group and the phosphate group. I'll give you a couple seconds to look it up, and then I'm just going to randomly call on a name.
Well, I know the phosphate, isn't that like structural support?
It is structural support. Yeah, and not always, but it is in the case of DNA structural support. What's the charge on it, Quinn?
Isn't it positive?
It's actually very negatively charged, I think. Wait, yeah, it's very negatively charged. I'm getting myself confused. What about the methyl group? Rachel, what do you know about the methyl group?
I'm sitting here trying to go back on my notes.
They're non-polar.
They're non-polar, you're right. What is this? Oh shoot, is this Asante?
No, this is Michaela.
I'm sorry, Michaela, it doesn't show your name, it just shows iPhone. So they're non-polar. So the phosphate is positively charged and the methyl group is non-polar. That's important because the acetyl group is negatively charged. If the phosphate is negatively charged, that's involved in this DNA backbone that connects one DNA base to the next, and the acetyl group is negatively charged, are they going to attract one another or repel one another?
Repel.
They're going to repel one another. The result is that this DNA is more open for expression when it's got acetyl groups added to it than when it has methyl groups added to it. The DNA is much more open, which allows the gene to be expressed in the DNA. When it's not open, when it's got methyl groups, the methyl group doesn't affect the polar group, the polar phosphate group, the negatively charged phosphate group. And the result is that this DNA is more compact. If the DNA is tightly compacted and tightly wound, it's not going to be expressed. There's a lot of research right now going on looking at histones and the role they play when it comes to whether or not they get methylated, whether or not they get acetylated. And are there ways to reverse this? Because every one of your cells contains your full genome. It's capable of being any cell based on the fact that outside of your red blood cells, which don't contain your nucleus, all of your cells contain your full genome. And the only reason that one particular cell has a specific shape and structure versus another cell that has a different shape and structure is because some genes are expressed in one cell and not expressed in another. So what causes that? What controls that? It's the histones, and whether they're methylated or acetylated. I'm sure there's some exceptions, that there's other factors playing into this, but this is a number one factor. So DNA where the histones are acetylated is open and active. It's not easily visualized because the DNA, the chromatin that's more compact, is inactive and it stains dark. When a cell is preparing to divide, the chromatin condenses into chromosomes. And that's where you can really see this banding pattern where certain parts stain light, other parts stain dark. But the compaction into chromosomes prevents tanglement and breakage of chromosomes during cell division. You've heard of genetic disorders where they're missing parts of chromosomes. I don't know exactly how that happens, but I'm sure it's related here somewhere. I have a little niece who has what's called Williams syndrome, which means that she's missing part of chromosome number 7. It's also called Elfin syndrome, if you've heard of that. RNA. So RNA is formed from nuclear DNA. And there's 3 different types. There's transfer RNA, which binds to amino acids and is involved in protein synthesis from the mRNA sequence. There's ribosomal RNA that's part of the ribosomes. And there's messenger RNA that forms the sequence from which the protein amino acid sequence is formed. The sequence is read, and from the reading of the individual bases, or the 3 codon bases along the mRNA, the amino acid sequence is determined. Stop. So if I look right here, do you remember what are the base pairing rules of DNA?
A with T, C with G.
A with T, C with G. T, A, G, C. Okay. So for each A let me bring something else up. What's the difference in the base sequence of DNA versus the base sequence of RNA?
Your cell.
And what does it base pair with? What does it substitute for?
T.
Yes. So in the case of RNA, U, A, G, C. So U binds base pairs with A base, T base pairs with A, or sorry, A in DNA codes for U, T codes for A, C codes for G, and G codes with C in the RNA. So this is DNA, this is RNA coding. So each base along the DNA base pair codes for one RNA base based on the complementary base pairing. So, a bunch of bases. The mRNA that would come from this or be coded by this is U, A, G, G, C, C, U, A, U, G, G, C, C, A, U, right?
Right.
Now, the protein sequence, if you remember, there's 20 amino acids. Each 3 sets of RNA bases codes for 1 amino acid. So UAG codes for 1, GCC codes for another, UAA codes for another, UGG codes for another, and so forth. So each of these 3 bases is called a codon. So each 3 mRNA bases is a codon. And with the exception of 4 different codons, each of the codons codes for, actually with the exception of only 3 of the codons, each of the codons codes for an amino acid. So let's see here. Bear with me just for a second here. So one codon codes for start codon, which always codes for methionine, one of the amino acids. Each 3 codons. Code for a stop codon. So they don't code for anything. They don't code for a specific amino acid. They just code for the end of that protein being synthesized. It's the end sequence. So just to reiterate, each DNA base codes for 1 RNA base based on their complementary base pairing and the fact that uracil substitutes for thymine. Each 3 bases is 1 codon that can either code for the start codon, and the start codon is actually AUG right here, or it can code for 1 of 3 stop codons, or all of the other codons, code for a given amino acid, 1 amino acid. So with that in mind, the process by which DNA is encoded into mRNA is called transcription. That occurs in the nucleus. Translation occurs once the mRNA and the ribosomal subunits are out in the cytoplasm. And that's where the mRNA sequence that we were looking at right here, this would be our mRNA sequence right here. Here. When this is converted into protein, this process of converting DNA into mRNA is transcription. The process of converting this mRNA sequence and translating it into the protein sequence is called translation. So the overview, DNA codes for mRNA, and that the process of doing that DNA to mRNA conversion is transcription. mRNA is going to code for the protein sequence, and that sequence is synthesized via translation. Any questions? Protein degradation. Sometimes organelles, or many times organelle proteins outlive their usefulness and need to be degraded. These proteins can be destroyed by lysosomes. We've talked about those. Those are the ones that break down substances, kind of like the stomach of the body. Misfolded, damaged, or unneeded proteins in the cytosol can also be marked for degradation by the addition of what are called ubiquitin tags. If you know the word ubiquitous, which means to be everywhere or commonplace, that's where this name tag got its name. It was just found everywhere, ubiquitin. So misfolded and damaged or unneeded proteins can be marked with ubiquitin tags that mark it for proteolysis. We talked about hydrolysis. Proteolysis is just the specialized or hydrolysis of proteins. So proteolysis using proteases or proteasomes, which is a specific cellular enzyme that degrades small and large proteins. So the damaged DNA or unneeded proteins are marked with ubiquitins for degradation by either proteases or the proteasomes that contain proteases. Remember, ASE is always an enzyme. Proteases meaning proteins, enzymes that break down proteins. Some extracellular materials, are defined as any substance contributing to body mass that are outside of the cells. We have body fluids, interstitial fluid, blood plasma, cerebrospinal fluid, cellular secretions, intestinal and gastric juices, saliva, mucus, and serous membranes. The extracellular matrix is composed of proteins and polysaccharides that are often secreted by cells that function as a cell glue and a medium for cell signaling. I learned in cell culture a long time that sometimes I couldn't maintain the function of cells if I didn't plate them without a matrix. I was involved, I spent a year trying to develop a mouse liver cell culture and realized ultimately that without putting them on a matrix, I could not get them to function as liver cells outside of the mouse. So the extracellular matrix works as a cell glue and helps keep cells in contact with one another when they're both present. Embryonic stem cells are in various regions of the embryo that are exposed to different chemical signals that channel them into specific pathways of differentiation. So based on the signals being given or the signals received, let's say based on the signals received, these stem cells can differentiate into multiple organs. These chemical signals can be slight, such as a slight difference in oxygen, or slight upper increased levels of carbon dioxide or decreased levels of carbon dioxide. The cells will then release chemicals that once a certain cell is specializing and differentiating to take on a specific shape and function, they can release chemicals that will influence the differentiation of neighboring cells. Think of differentiation as being a specialization of the cell and its shape and function. And when we talk about cell differentiation, we're kind of talking the same thing about that gene expression, and that all of our cells contain our full genome, except for red blood cells. But they all contain our full genome, but based on what signals were received, that cell specializes / differentiates into a specific type of cell. So a ball of undifferentiated cells can, based on the signals received, can differentiate into multiple types of cells. Any questions? One very crucial process that's important in normal cell development and our immune system and so forth is apoptosis, which is controlled cellular suicide that eliminates stressed, unneeded, aged, or excessive cells. Damaged macromolecules within a cell can activate a series of caspases. Caspases are enzymes that are going to destroy DNA, cytoskeleton, and so forth. So it's a programmed self-destructive suicide. The cells shrink, detach from one another, round up, and start blebbing, meaning to just break apart. Little pieces are sloughing off. The dying cells release chemicals and apoptotic bodies that stimulate macrophages. If you remember, a macro means really big. Phage means a cell that can perform phagocytosis. So the dying cells release chemicals and apoptotic bodies that attract the macrophages that then phagocytose those dead cells or dead parts of the cell. Cancer cells can avoid apoptosis. Remember, that has to do with cell identity markers on the cell surface. However, heart muscle during heart attacks and nerve cells during strokes will undergo apoptosis based on the fact that there is an oxygen deprivation signal of hypoxia. Just say one more thing here. Sometimes apoptosis is, sometimes it's like up here, it's needed. The reason that your fingers are separate and you don't have webbing in between your fingers is based on the fact that there's a programmed cell death, apoptosis, that occurs that separates your fingers by killing the cells that connect them, or not killing them so much as having those cells die. Hyperplasia is an accelerated growth. During anemia, bone marrow will undergo hyperplasia so that red blood cells are produced at a higher rate. Atrophy is a decrease in the cell size of an organ or body tissue. So it can be cell number and size. So hyperplasia is increased size or number. Atrophy is a decrease in the size. Cells can wear and tear. because of cell aging due to chemical insults and the formation of free radicals. Remember how free radicals are produced all the time. Most of them are neutralized via the peroxisomes, but they do still and can accumulate over time. And the result is that the cells age. Energy production is diminished in radical damaged mitochondria. Temporary lack of oxygen causes blood vessels to clog with fatty materials, leading to accelerated rates of cell death. Remember, if you have, let me put it this way, if you have fatty clogs in blood vessels, well, what do you need blood vessels for? For oxygen transport and carbon dioxide removal. Well, if you have decreased oxygen flow, you're more likely to deal with apoptosis due to hypoxia. Hypoxia is reduced oxygen levels. One last thing that's involved in cell aging are telomeres. Telomeres are found on the ends of the chromosomes, and they protect the chromosomes from fraying or fusing with other chromosomes. One of the theories of cell aging is that there's a telomere clock that determines the number of times a given cell can divide. Each time the DNA is replicated for cell division, 150 to 100 of the N nucleotides are lost and the telomeres become shorter. When telomeres reach a certain minimum length, the cells stop dividing. So the length of the telomere is the telomere clock. Once they reach a certain length, the cell stops dividing. Immortalized cell lines, we have immortalized cell lines that just continually divide and divide and divide and divide and divide. And embryonic stem cells can continually divide. Their telomeres don't change in length. Just to bring this last point home about telomere length, if you're familiar with the disease of Hutchinson-Gilford progeria syndrome, which we've probably heard of progeria. You might have heard of it. This is where kids prematurely age. One defect that they found, one genetic defect that they found with children with progeria, is that they have mutations in a lamina nuclear protein that's involved in this nuclear membrane protein, lamina. And the telomere length of these lamina are shorter than those in normal age-matched controls. So the result is that the lamina protein in these, in progeria hemopoietic cells, which are cells that are involved in producing bone marrow and red blood cells don't express the laminin A with normal length telomeres, so those cells stop dividing prematurely. So their bone marrow cells stop dividing before they should. Based on, and it's because of the fact that they're normal, it's hypothesized that it's because of the fact that the telomeres are shorter than they should be. Any questions about chapter 3? Let's take 10 minutes and come back and start chapter 4. Not chapter 4, chapter 7. So I will see you back here at 11:08.
Are you gonna stop the video?
You know what? I don't think it's making a difference. I can stop it. I'll stop it, but I don't think it's making a difference. There's something else going on. And
okay. I'm just asking. I didn't know if things were working out or not.